making kind of a work. I'm beating my own drums at this starting point itself, but I hope you see why at the end of the talk. Okay? So this particular poster was prepared by Vivian, a former student of mine, and this talk is about science, the way of doing science, how I started off. It's going to be a personal voyage and all that, and I'm trying to make it interesting for you students, primarily and research scholars. Of course, teachers are also here. The topic of my presentation is, why do we need oxygen? Why do we use oxygen? So this seems to be pretty, kind of a redundant kind of a question to us. People think they know. It's been, this kind of a question should have been there around for quite a long time. People have worked on it. I say that we got it all wrong at the prima facie outset itself, I would say that. And I will now try to answer this question with a very radical answer. So now the contents of this talk would be the story of murburn concept. Murburn concept is the explanation that I am putting forward to say how oxygen is used by our body. And then what is wrong with the prevalent hypothesis or theories. The prevalent hypothesis is called electron transport chain, E. P is proton pump, C is chemiosmosis, R is rotary ATP synthesis. So those of you students who have studied this, you know these are the 4 elements of the currently available paradigm. So that's the paradigm for cellular respiration, the most fundamental process that all oxygen utilizing cells use for availing energy. Next, how does murburn concept explain mitochondrial oxidation phosphorylation, that is represented by MOXphos, I will call it. And then the projection. So primarily you will see that the talk is divided into 3 parts. First about my story, next about what's wrong with the current understanding, and how my concepts explain. Okay. So before I start off with the talk, let me tell you, I look unconventional, I speak unconventional. So let me explain why such a thing. You will see that my talk will be filled with I, me and mine. Please bear with me. Okay? You will see. It's my personal, passionate, professional pursuit. I believe that this is required for science at this moment, and that's why I am adopting that stance. Please tell me if you don't think that is right at the end of the talk. Okay? Then there will be too much text in certain slides. Don't worry about the text. I am the beautiful guy here. Please look at me. I will guide you through it. There will be sometimes very fine prints. They are not supposed to be read. They are there just to show you certain things. And then interruptions. If I am saying something wrong, if you teachers would not want your students to be informed of something wrong, teachers can interrupt me anytime. The students, I would request you, if there is a doubt that you think, oh, I have to clear it right now, please feel free to interrupt me. Otherwise, I would request you to keep a note of the slide number and ask me without at the fag end, because I will be available. I'm all here for defending my post. So, before I start off, let me just tell you or ask you something about practice of doing science. You all do science, it's an experimental way of doing works. So, let's say we are weighing an animal. We are life science people. We are weighing an animal. I say I took an animal, put it on a weighing balance. 38.9 milligrams. Okay. Second time I took another animal, 40 milligrams. Another time I took another animal, same species, same, and then 40.1. Like that, 38.9, 40. So I say some guy who did that, he writes the animal weighs 40 plus minus 0.2 milligrams. So can anybody here say something about the accuracy and precision of the experiment, particularly from the student side? I told you 39.8, 40.2, 40, like that. Many times. That person got 40 plus minus 0.2 milligrams. Can you say whether the experiment has been done accurately? Was the data precise? Do you know what these things are? Okay, can anybody tell me? If you don't want to speak, it's okay. This is very important for this talk because this has to do with the core way in which science is done. We are dealing with two concepts called accuracy and precision. Now, when I say it's 39.8, 40.2, 40, and then I say 40 plus minus 0.2 milligrams. Is it accurate? Is it precise? Can you say whether it is precise? Say something. I would say as an experimental scientist, it's okay. It's precise. But in terms of accuracy, can you say that whether it is accurate or not? So, this is something that you need to find out. What is accuracy, what is precision? Because science has to do with pursuit of both accuracy and precision. Experimental methods allow you to do something with only precision. Okay? Accuracy is called by the scientist. Never by no experiment, no instrument can tell you whether an experiment is accurate. So, science is a very subjective pursuit.
But you are dealing with an average.
Yes.
You are not dealing with a value.
No, I will tell you now. Very good. Very good, madam. Now, let me tell you why it cannot be accurate. Now, if I tell you the animal is an ant, then the value can be accurate. But what if the animal is an elephant? The accuracy goes up. It's not right. It's not approaching truth. So, with this I want to start off my talk. This we will come back later.
I
Yes.
feel there's a conceptual problem here because the moment you're talking about accuracy or you're talking about an individual value and what you are citing is an average value. An average value, what you are doing with that plus and minus is giving you an indication of the accuracy of an average value.
That is what I said. It is precise. It is not necessarily accurate. I said accuracy can only be called by.
If the instrument is standardized, then why it is not accurate?
Because I said now I'm giving you an information that if the animal
Weighing machine, you are weighed into a weight of 65 kilograms.
No.
Don't you believe so?
No, the point is an instrument will only tell you certain things. After that, the interpretation comes from the scientist. So now I gave you one data. The animal is an ant. Ant will have 40 milligrams weight, but elephant, it might not.
You cannot use the same instrument.
That is what I am saying. I did not give you that data. I did not give you that data. You did not understand. That data I did not give you. I gave you only some data. In real science, most of the experiments that we do, we don't know what are the variables.
I think the example you are quoting is not correct.
Okay, we can come back to that later. No, you very valid what you think. But I am very clear what I gave. I did not give you the data about what the animal was.
So maybe you are trying to say that it's an incomplete report of the data.
Exactly.
Not
So
the data is not accurate and not precise. The incompleteness in the reporting.
the inference is you cannot say anything can be accurate. Accuracy is called by the scientist. That's the whole point. That's my argument. You did not get that. We'll come to this later. Because now when you start an experiment, you come to a conclusion of accuracy. You try to seek accuracy. That accuracy is something that will always evade you because you don't have information about everything under the sun. That's the whole point I wanted to give you that. I can give more and more data and make this more accurate or not accurate. Okay, now let me come to the first part. Okay, now I'm basically an enzymologist. Enzymes deal with proteins. I took proteins because proteins interact with all kinds of molecules, and they are like something like actors. They are the main protagonists of life. Of course, nucleic acids. There are other polymers also, but they work from behind the screens. And nucleic acids primarily interact with proteins only. So, since proteins are the major protagonists, I took proteins as something that I was interested in. I was educated in, facilitated by Kelath Murali Manoj, our former president. He was then the Science and Technology Minister, and he brought the national level Science and Technology exhibition to NSS College at Pal. where I did my PhD. So there I came to know about biotechnology. I wrote the entrance and I got into it. I took Goa University as my first choice. Of course, Goa was a fun place. And I thought marine biotechnology, I'll be doing scuba diving and all. It happened to be something else, okay? So now I'm trying to tell you how chance takes you to certain areas. You know, this is something that I wanted you to know, because now you're doing your MSc, you are doing certain things you don't know, certain things you have some ideas. I want you to have some idea of metacognition. What you do, you are in a kind of a cycle wherein you are supposed to submit assignments, do tests, this, that, you know. So you should be always conscious of what you are doing, what you are learning, how your career is shaping up, how your interest is being pursued. That's what this part of the talk is about. So then I did my PhD in IIT Madras. That was after some stints in JNU. I didn't like it. Central University Hyderabad, I didn't like it. Then I went to IIT Madras. I liked the ambience. I did my PhD there. And in my PhD, the biggest thing that happened to me was one of my friends died when we had gone to the beach. So while trying to get his death certificate, the administrative ambience of our country was such that they expected bribes for even a young man's unfortunate death. So I was very shattered. I was a scientist and all that, but then the hero in me wakes up and says, really will change the corruption in the country. I will come back to the country and fight the corruption. That's what I decided. But before that, I had to prove myself that I was a good scientist. So I went abroad. So I got into a European Union fellowship, and then you will see all my research was always about proteins like proteases, lipases, epoxide hydrolases. They're all enzymes, okay? So now my fundamentals in this area was, when you do reactions, you do collision theory, okay? Then you have transition state theory, wherein there's a transition state acquirement, and then you have product formation and all that. These were the fundamentals. And when you come to enzymology per se, you have Fisher's lock and key, where there is tight affinity, predetermined tight affinity between the enzyme and substrate. And then you have induced fit, wherein the enzyme changes the conformation upon the availability of a substrate, right? And then the standard Michaelis-Menten kinetics. This was my world. I guess this is your world too, right? Okay. Now, this is what I was into until now. My world changed when I went to University of Illinois, Urbana-Champaign. I went to the lab of Lowell Hager, the best professional mentor anybody could have. He was National Academy of Sciences member, 25 years chair of the department, and the best person I have met. You wouldn't imagine we would hug, kiss when we part. That kind of proximity I had with this man. So, before starting the crux of this talk, I want to pay my undying gratitude to this great man. He passed away in 2014, but he lives on in my memories forever. So with that, I want to say, my life, he introduced me to heme proteins, redox enzymology. And that's what this talk is going to be about. So I came through a lot of things, finally settled down to something. This is what you people also will have to do. You will see a lot of things, and then you can't go chasing everything and everything. Like you marry one, you choose one topic like that. You have to settle. You have to not settle. You have to focus on certain things. That's what it is for. Now, this slide is put there because currently my mechanism is the standing one for cytochrome P450, which is the enzyme system that metabolizes majority of the drugs that are metabolized in the human liver. Okay? Now, it is a $1 trillion industry. My paper was published in 2016. I had to go through a major story for that. It will come in that. But then, that is what cytochrome P450 are. You have to stick here somewhere? No problem.
There's a point.
Is this the pointer? No. This one just.
Yeah.
Oh, this one. Okay. Okay. Cytochrome P450. Then chloroperoxidase was the enzyme that Lowell had discovered in 1960s. So that is here CPO, called CPO. They are both heme thylate enzymes. Heme is this, thylate is the sulfur ligand here. It's cysteine. So unlike all other enzymes, these enzymes are the most maverick enzymes that you can ever find. They do the most diverse types of reactions. They will have very diverse enzyme substrate interactions, no specificity. You can see epoxidation, sulfoxidation, hydroxylation, so many different types of reactions they will do. These are the common reactions. Chloroperoxidase will usually chlorinate certain molecules also besides that. Cytochrome P50, which will be called CYPs, they are the enzymes that metabolize the drugs and other xenobiotics that enter our body. So, my mechanism is the standing one in the world as of now, and that is because of my experience with chloroperoxidase, which Lowell introduced me to. When I started working with him, as some of you would do in PhD, postdoc and all that, you would be given some topics to work on. You would have to, there would be a funded project, and you have to work on along that project. You just can't do any fancy thing that you want to do, So that's a reality that we all have to live with. So I also went there. In all the other places, I worked like that. But my PhD, I took pretty much what I did, I chose. So here I had some NIH project, some interactive collaborative project, US Navy project, so many other projects. So in all other places, I worked for 5, 6 years, 7 years. Papers were coming trickling by, 1 in 2 years. I go to US, it comes like that. In a year, 3, 4 papers just came like that. So in the first year, I could get very easily some papers. They were not very interesting for me. This paper is a PNAS paper. I wanted this paper to be retracted, and in fact, the paper that I am really happy about is in the second year. I talked to Lowell. I said, Lowell, I'm retiring from science. I'm going back to my country to do social work. Why would you please give me some freedom? I want to do things that I want to do. He said, feel free, he said. And that 1 year, I sent back my wife. We had a kid then, I sent back my wife. We can do that in English, you can use whatever pretty much you get. So, and then I started working on what I thought were very, unresolved questions in the field. So, this paper resulted first. Here, what was happening was, when you give chloroperoxidase into the reaction, after some time the reaction stops. People thought that peroxide inhibited the enzyme, because peroxide is a reactive species and it can modify so I did certain things and I said, it's not peroxide inhibition, it is peroxide depletion. Very simple thing, very small thing. What people thought and what I saw, it was different. And I challenged. Okay, so I said that this enzyme can turn over millions of peroxide molecules without getting deactivated. And it has got a very high catalytic activity. Catalase is peroxide depletion into water and oxygen. So that is called catalytic activity. So that it has got very high catalytic activity. So this one was one paper. But the paper that I was so passionate about, which is the paper of my life, I would say, is this. It's a paper that I published in 2006. I opposed Lowell's mechanism on chlorination. It was the 40 years standing mechanism. He said that the enzyme, the chlorine intermediate is within the active site, the substrate goes in and it reacts with the enzyme at the active site, and the chloride atom is transferred there. Now, why did they say that? They said because when you give this enzyme a mixture of substrates A, B, C, D, E, the enzyme chooses only say C. Okay? Now, if you give it to a diffusible species, some reactive species like bleached, HOCl or something, calcium oxychloride or something like that, it will react with A, B, C, D, E, all. And not only selectivity, specificity. So now if you have a phenyl ring, ortho, para, meta, it can attack at many places. The enzyme reaction shows only one place, but the diffusible species shows multiple places. So the enzyme reaction was showing, and that was one of the reasons. There were many other reasons why which they said that it was active site. I showed in this paper that for the first time an enzyme produced diffusible species can mediate both selective and specific reactions. So for the first time, all the active site, induced feed, all that ideas, you're going beyond that. I had difficulty to get Lowell's name in the paper. He said, no, I'm not putting my name in that. So later on he came to accept my ideas, but I was too stupid and too bullheaded, and I said, no, you said earlier you won't put your name, so I put it, and then you said, okay fine, he has got so many papers, so for him this is nothing. So I had a tough time. Finally it got published after my 8 years in BBA, okay? But this paved way for very fundamental difference. If you see this paper, the last paragraph of this paper discusses a very important key concept in terms of metabolic perspective. Now, in the same year, what had happened was, in 2001 I had come back after working with Lowell. Then I did 3 years of social work in Kerala. Satyam Jetu, I started my trust, and I didn't become Atal Bihari Vajpayee or Prime Minister. Nothing happened. No success. All my money ran out, and I had to go back to get some money. I went back to US, and I started working once again on heme enzymes. And in 2006, I was working on cytochrome P450s, and I was given an NIH project in Washington State University, and there I was supposed to minimize. See, when liver enzymes metabolize xenobiotics, you find lot of peroxide and superoxide production. So when you try to reduce when you want to have a good drug, you don't want reactive oxygen species. You want to minimize them. So my project was that. So I told my PI, my boss, your project is not well defined. Your targets are not right, because if I take reactive oxygen species out, the reaction ceases. The reaction requires reactive oxygen species. You cannot have the reaction, so your project is ill-framed. He said, please move out of my lab. So I had to move out of the lab. I went to another lab. He was very nice, he moved me out of the lab in 3 months. So, so that was. So I came back to India, started my own group, but before that I published it in Cornell University's RSA in 2006. The same work, it took me 10 years to get it published. But in the meanwhile, I could get the 1 electron reactions of chloroperoxidase. I could get Lowell to put his name. There is the chlorination reaction. That enzyme does a lot many reactions. So in that, I got 1 electron reactions. I got Lowell to put his name for the 1 electron peroxidations. Here we said it is not in the active site, it is somewhere outside on the enzyme. For that, Lowell put his name. So you see the citations and all are going up. Credibility goes up. I am trying to tell you a lot of messages in this. And then when I came back to India, like what Dr. Ajit said, I was a professor at VIT, PhD, Shastra. I had some students, Dept and MTech and PhD and all that. And in a matter of 10 years or so, I published a lot of small papers in PBRC. It's not a bad journal. It's got 80% rejection rate. So yeah, it's a very decent journal. It rapidly publishes. So I got some papers, and finally this the major story, I could publish this one in Biochimie in 2016. This gives the overall picture of the whole reaction system, the complex nature of the reaction system. So what I did is, the elephants hand is like this, trunk is like this, the body is like this, different parts I published as different this thing. The heme enzyme is relatively cool, you can publish. When you come to the SIP system, that's very competitive. It's a very vicious field. People won't allow you to publish things that go against their doctrines. Because why? Because it's a $1 billion industry. They optimize drugs based on these ideas, and when you try to change fundamental ideas, they don't like it. So, but yet some new journals like Plasman and all those, they were getting released, and I published there, and this was a very fundamental paper wherein I showed until last year the electron transfers between proteins were supposed to be through protein-protein interaction, long distance protein electron tunneling, it was called. It's the person Rudolf Marcus, the Nobel laureate, his theory was applied. I showed what I did, a very simple reaction. Took one protein in dialysis membrane, one protein outside, and I showed that even when they are separated by a dialysis membrane, you could get electron transfers. So with that, I showed that electron transfers can occur without protein-protein interaction. Enzyme to substrate interaction need not go through active site. So now we have got a very different paradigm. Okay, this paradigm was established. But for the SIP system, I could not get my whole paradigm in. So in 2015, I went to US, spending my own money to present these works in a lot of conferences. And there I met one person, Cruciani. He is a big name. If you want, you can Google him. And he was making a talk on enzyme substrate interaction, drugs, and all that. And I asked him, don't you think this is far-fetched? And in front of all the people, he acknowledged. He said, these molecules don't look like they can go into the enzyme. And I'm just doing the simulations. This is what people believe, he said. And he was very interested in the ideas that I had. So, in spite of having all the experimental data, I had to put a theoretical paper first, wherein which I introduced murburn concept. Here we are coming to the crux of the talk now. Now, murburn means mured burning. Mured is closed burning. Burning is any reaction that you would have with oxygen, right? So, it's just anything that burns is a reaction that uses oxygen. Now, it is also mild unrestricted burning or molecule unbound ion radical interaction paradigm. So this was published in silico pharmacology, wherein I introduced the theoretical aspects, and then just when I was about to give it up, I had spent all my savings of US, all my salary, everything in this pursuit, and I started like this, and just when I was near the jackpot, I was about to give it up, when finally it was accepted in Frontiers in Pharmacology. You see, murburn concept explains the metabolism of xenobiotics in hepatocytes. It's been published for 1.5 years. I've got only one citation for it. Only one citation. Where in this paper, I have compared the erstwhile hypothesis and my hypothesis, and I have listed 50 reasons why the erstwhile hypothesis is misplaced and why my hypothesis is right. But people will not, they will pretend they have not seen it. That's the beauty of science. And that's the first, if you had seen Max Planck's citation that I had written in the first slide, scientific truth doesn't become, because accuracy is always upon scientists. I can choose to see what I want to see. That is what I am trying to come to. Now, here is the murburn concept earlier. The substrate binds, then the spin changes. These are all details which biology students might not be interested. But the point is, you see here the substrate xenobiotic. If you see diverse kinds of substrates, xenobiotics are supposed to bind to liver enzyme. Do these enzymes know what kind of molecules are going to come? Suppose lipase, carbohydrates, proteases. They know evolutionarily. They know what molecules are going to come. But xenobiotics, they don't know what active site, they don't have any idea. So here these had such kind of mechanisms, and they said that reactive oxygen species was a side reaction. I said no, the oxygen activation was supposed to be done by the heme enzyme. I put the horse the other way around. I said oxygen reactivation was done by the flavin enzyme. There are two enzymes in the liver system. One is a hemoprotein, one is a flavoprotein. So now reactive oxygen species is produced. This is stabilized by the heme enzyme, and then you have the reaction. So this is my hypothesis, very diametrically opposite. And now you see the mechanism. This is something that you can understand very easily. Please see here. This is the current hypothesis. Substrate comes, binds, induced fit, it goes, reacts, comes out. My mechanism is not that. An intermediate is produced by one enzyme that can go and bind to the heme enzyme, get stabilized, and the second substrate, it can be bound anywhere, non-specifically or it can be around. So this diffusible species can come out of the active site and react anywhere in or around the enzyme. So Michaelis-Menten kinetics don't hold. Fisher's lock and key theory don't hold. Boschland's induced fit theory don't.
But it is a bisubstrate reaction.
It is a kind of bisubstrate reaction.
So the one substrate is going to the active site of the enzyme.
Right,
The
but
oxygen species is a substrate.
correct, one kind of substrate. But you should see the ultimate kinetics of the reaction is governed by this point interacting with that, and that interaction is somewhere around. We are interested in this molecule interacting with this molecule. We are not in, see, earlier treatments were enzyme plus substrate complex, ES complex, transition state. That is not what is important here. Here the reaction of the DROS with these kinds of molecules are of the drugs. That's a very good question, but that is not the...
In bisubstrate enzyme reactions, any A or B can bind first.
Yes.
There are sometimes some substrates will be preferred to the other substrate.
No.
That is a here oxygen, active oxygen species is preferred. That's all.
Dr.
No,
Sharif.
it is my case is
No problem. Dr. Sharif, the point that you are not getting perhaps is that here earlier these molecules were supposed to be interacting with the enzyme at the active site. That is not the case. That is the whole point. And these are the molecules of interest. And this reactive oxygen species was supposed to be formed here. It is not formed here. It is formed by another enzyme. That is the whole theory. Yeah. So now the whole scheme, scheme of reaction can be, this is the mechanistic scheme. Please don't worry about it. I am not going into the detail. This is the mechanistic scheme. This is the kinetic scheme. This is the stoichiometry scheme. So the new hypothesis gives a complete different picture of the kinetics, mechanism and stoichiometry, reaction chemistry. So, now what is the application of this? Now I am coming to the end of the first part in a short while. Here you see, these are the different types of drugs. Ibuprofen, warfarin, say for example, ritonavir, sildenafil, Viagra, all these kind of drugs are metabolized by a single enzyme. Single enzyme. CYP2C9 it's called. Okay. Now, with a single enzyme substrate binding and active site, you cannot even imagine this kind of a mechanism. My mechanism explains that.
So it's actually a mono-oxygenation process, isn't it?
Yes.
Oxygen is actually the one oxygen atom from the molecular oxygen is added to the
Mixed oxygen.
To this,
It's not a classical oxygen, it's a mixed oxygen. Yes.
Yeah, mixer function of oxygen.
Yes.
So one oxygen atom is transferred to the xenobiotic.
Yes.
And how you are telling that it is against the Michaelis-Menten?
This is Michaelis-Menten. The substrate binds and the substrate interacts at one place. That is ES complex. Here, this molecule need not be bound at all. It can be freely diffusing. It is not at one site, multiple sites. So kinetically it will not be reflected. You will see, because of that, now for example,
Is there any proof for that?
my paper is all about that. I have experimentally proven that.
It is binding at the other sites like serologic studies.
Pardon?
Any extracurricular studies that they...
If you see the two papers, yes, in silico pharmacology, my paper, and if you see my the frontiers in pharmacology paper, they have reasoned out n number of observations that other people have published. Crystallography, all enzyme, so fluorbiprofen complex CYP2C9. The distance between the ion center and the fluorbiprofen is 10 angstrom, 9 angstrom. For bond formation, you need 2 to 3 angstroms. You cannot explain that. So, very simple. All the data currently that is available abides by this, my hypothesis. They were not able to explain whatever they were getting. My hypothesis explains practically everything that is there. Until now, I have not seen anything that my hypothesis cannot explain. If there is something, I will change my hypothesis, or my hypothesis can be changed. That is what scientific pursuit is. So, this is not microelement kinetics, and that is precisely what I showed here. See, now in substrate, you see in CYP2C9, what you get when you have a, what is a microelement kinetic? You have a hyperbola, right? Typical hyperbola. Right? You know what a hyperbola is, right? You have a first order kinetics at a lower substrate, a zeroth order kinetics at a higher substrate, and somewhere in between in the middle. So that's a hyperbola. Now we'll come to that after some time. Now, if the same thing can be given by the diffusible species interaction with that, and not only that, if you see, after some time, the CYP reactions, they come down if you increase the substrate concentration. With so many substrates, with so many CYPs, this was the observation. Now
that is because the enzyme may be limited.
No, enzyme is getting inhibited by the substrate. That is the explanation. That there are multiple binding sites for the substrate within the enzyme. Suppose I will tell you 1 second, Dr. Sharif. If you have one substrate, it goes. When you have more substrate coming, this substrate is bound. A second substrate comes and binds, thereby blocking this molecule's exit. So that was the Michaelis-Menten explanation for substrate inhibition. That was the explanation for substrate inhibition for that. But then, Occam's razor, there is a term in science called Occam's razor, in which you will have to imagine that suppose if all these molecules have multiple binding sites for multiple CYPs, whereas my hypothesis says it's very simple. This 2 electron reaction has got a 1 electron intermediate that collapses in different ways. And I could, in my class 1 paper, I have explained, and this was selected for presentation as an oral speaker in international conference in cytochrome P450s. So that's a very well established thing, substrate inhibition part. That's very well established. My hypothesis explains it very clearly. So, and the most important part of it is, you know, now this paper is under consideration. Have you heard of something called hormesis? Hormesis or idiosyncratic dose responses? Hormesis is like this. Now, if you increase a dose, what do you get? You concomitantly you get increase in response, right? In biological systems, that's what you know. But
it is not always.
Not always. Correct. Not always. Yes, that is one time when you get when at dilute concentration, when you get higher response, that is called hormesis. Okay? So currently there are no very solid explanations for that. My hypothesis could provide one of the explanations, solid mechanistic explanation, because radicals are stabilized at low concentrations in platinum. Thank you. So this is one of the explanations for hormesis. So it explains the promiscuity of reductase. How can one reductase transfer electrons to hundreds of different CYPs of different topographies? If you say superoxide is the electron transfer agent, then you can explain. So like that, it explains many things mechanistically. So now we come to the crux of my talk, which will not. Now I have showed you in a comparative system of simple enzyme called chloroperoxidase, which had one heme enzyme, peroxide, where the two react. In the cytochrome P450 system, there are flavin enzyme, heme enzyme, and cytochrome B5, then NADH, oxygen, so many. It's a complex reaction. I have shown in a simple reaction setup, relatively complex setup. Now, mitochondrial oxidative phosphorylation is a whole different reaction system. It is a complex thing that happens in a cellular organ. Now, this talk that I am going to talk to you about, probably you are studying this in the first semester. When I had to teach it as a professor to my students, I found it very difficult to teach this. I always used to say, this is wrong. I think this is wrong. I think this is wrong. Always I used to say. And I found it very difficult to teach. And when I submitted one of my papers in 2010, Stuart Ferguson, he has written the most popular book on bioenergetics now. He was the editor for that paper. He said, well, Dr. Murali, you say that based on some simple observations, you say you are putting down a whole theory. That is not correct. Even I got some arguments against Mitchell's chemiosmosis hypothesis, but there are so many observations supporting that, so I cannot pull it down. So he had said that in 2010. To that I had responded, you might reject my paper on this thing, but I think that chemiosmosis hypothesis is wrong. Please publish your work. He did not. He is still a defender. And it so happened that I came back to this. I was a principal of a college in 2016. I lost my job. And then I thought, I will sit and do this. I will break this explanation. Because I thought my mechanism can explain this better. So this I wrote in 2016 and it was accepted but retracted. Because there is a lot of political pressure. Now, do you know what the current hypothesis is for this? I'll just show you. Oh, you don't have audio, so I will skip I think I will skip all this. Okay, so now this is you remember seeing this, my students? Complex 1, complex 2, complex 3, complex 4, these ATPase and all that. So currently NADH is supposed to give electrons to complex 1, and then coenzyme Q, complex 3, blah blah, it goes through like that in a very nice coordinated way, like a very good, nice circuit and all that. And all the while protons are being pumped out, and complex 1 equation, all that, all beautiful, dandy, all textbooks, Leninger, all these things say this. Okay, so you know this. Okay, this is all wrong. I can tell you very clearly. Now, what is it like? This is like something like this. You imagine protons, they come in. Please, I have come here to say this. This is very important. Please have your attention here. This ATPase can be compared to the turbine here. When water comes in through the penstock, this is like a dam. Now it goes round and round and makes the energy currency power line. It can be compared to something like that, or it can also be compared to a car, wherein fuel is burned, NADH is oxidized in the engine, and then the alternator, recycles and stores it in the battery. Right? Battery can be similar to ATP, like that. So the comparison can be in structural and functional modularities. You can compare it like that. What is wrong with this? Now, first, as biologists, just think as biologists. You can very clearly think very, very simple ideas. Now, currently, this mechanism is too fastidious. Imagine a thing that evolves. At the time of evolution, you need for ATP synthesis, it needs a gambit. You know what's a gambit? Have you played chess? Gambit? You have to give a smaller piece to get a bigger one. So proton is being pumped out to get something, and you have to have n number of things arranged in circuits and all that. It's It's a very fastidious requirement. Evolution doesn't work like that. Of course, our complex eyes can be formed from very simple rudimentary parts. But if you study this, practically every single element is non-dispensable. That is, and it's a gamete system. Gamete system don't work in evolution at the beginning stage. And this is supposed to be the core energy forming mechanism of all life forms. Only if this is there can life do anything. Okay, this is the fundamental thing. Okay, and then it's a very low efficiency scheme. It's got only 40% efficiency. Why? Very importantly, this is the most important thing. You know that isolated complex 5, it has got only an ATPase function. It doesn't have ATP synthase function. Why would nature make an enzyme to make ATP when it hydrolyzes ATP? Imagine an enzyme does both reactions, this way, that way. Will it serve a purpose in real life? Just imagine. Of course, there are redox enzymes that catalyze this way and that way, but they have certain operators, regulators. But these don't have such kind of mechanisms as much as we can see. So, the most important thing is complex 5 has 10 to the power 7 times more affinity for ATP than it has for ADP. Why? You would imagine that a substrate will have more affinity, not a product. Product has to dissociate with from the transition state. It's a very simple thing. Of course, Paul Boyer has said rotate wave, energy is put and the affinity changes. I think that is all stories. Now, the next thing is oxygen. In the current hypothesis, you know what is oxygen's role? It just sits here waiting for 4 electrons and 4 protons, happily bedded to complex 4. Give me 4 protons, 4 electrons, I will sit here. Why should oxygen sit there? Why? Oxygen is happy, it can move around. Why should it sit there? And, you know, superoxide, peroxide, it is found plenty in mitochondria. It is documented. So, there is evidence that reactive oxygen species are formed. So, it is just a aesthetic notion that, oh, it just sits there. So, it puts a lot of, now if I show you the calculation, you might not like it. There are just some calculations which say that, the oxygen can, move approximately 10 to the power 3 angstrom in microsecond time scales within that. 10 to the power 3 angstroms within microsecond time scales. It can easily, oxygen is a diradical. You know what a radical is, right? It has got radical, has got an unpaired. Oxygen has got diradical. Now there are some catches, skin catches, there are triplets, singlets. I don't want to go into the concepts now. You can deal with it if you are interested after the talk. But the point is, this oxygen can easily, in presence of metal centers, it can easily get into singlets, triplets. There can be so many different species formed. They can all get interconverted. So, oxygen will mess with all the circuitry. There is no circuitry. You cannot see coenzyme Q. It's a lipid soluble free small molecule. It is supposed to be taking from one place and giving to another place. Why, Baba? Why should it do that? There is no rule. Oxygen, so coenzyme Q, cytochrome C. It's supposed to take from one protein to another protein. Why? So, these are the questions. Then stoichiometry, different stoichiometry. Per oxygen atom, how much phosphate, per hydrogen proton, how much phosphate. All these ratios changes. And they are not integral values. Then, very important, this is very important. You will find this very important as a biologist. You know cyanide? Now cyanide is supposed to kill human beings by virtue of binding to the complex 4. It's supposed to block oxygen. But you know what is the KD of binding of cyanide? It falls around millimolar levels. Now if you want millimolar levels in your body, you need to eat grams amount of cyanide. had shown heme enzymes can be inhibited by cyanide by other ways in a diffusible reaction paradigm. I had shown. And that KD comes in sub-micromolar ranges. That mechanism. So, when low amounts of cyanide kills, it cannot be by a binding based mechanism. It is by a diffusible species based mechanism. So how can cyanide kill at such a low concentration? That is something that current mechanism cannot explain. And then uncouplers. This was supposed to be Mitchell's big success. Uncouplers, they don't really explain. I am not going to go into this because they require great understanding of the subject. This is something biologists can easily understand. Now suppose take the structure of complex 1. Now, if you see the ion center, the electron transfer, the wire, this is supposed to be like a wire. It receives electrons here from the NADH and then it is supposed to go through like this. Okay? Electrons are supposed to go like this and protons are supposed to get pumped here. The problem is, there is no connectivity between the electron transport and proton pump. How does this protein know? There is no connection. And these distances are too far for functional electron transfer. And the redox potentials, from negative value to positive value you can transfer electrons. Many of these are not like that. Negative to still negative. How can you explain that? Well, they say, well, it can be some mechanism. They don't have it. But my mechanism, you will see that it explains all this. Why it is like this? It's a natural ploy. It is a natural ploy. And then, why is the distribution ratio of this enzyme, complex 1, low compared to complex 4? It was the same thing in cytochrome P4 reaction system. So many the projections. If you have these projections, you will have reactive oxygen species formation more. Why did you not have it here? Why was it not there? Why is it like this? Why should it come 15 angstroms, 15 nanometers out? Why? These are not explained by the current hypothesis. Okay, so no direct evidence for any proton pumps. Direct evidence is lacking. So now is the most wonderful part of my talk, and this is what I want to tell you, my dear students. When I go to my class, the first thing that I say is, if you find any fault in me, you get full marks for assignment. If you ask me a question that I cannot answer, you get full marks for assignment. That is for the fundamental aspect that you need to start questioning things. I asked a simple question to myself. How many protons are there in a mitochondrion? Can you make a calculation? It's a very simple thing. Mitochondrion is 1 micron in length. It is a cylinder, pi r squared h volume. Just put the value. You know what is the pH? 10 to the power minus 7, 7.4. You put all that value. You get how many? Less than 10 protons in a mitochondrion. You know how many complex 1, complex 2, complex 3, complex 4? Tens of thousands to lakhs. Do you know what is the, HO bond breaking? What is the value? It is 460 kilojoules per mole. Now, there are 10 protons, lakhs of proteins. Do you think these proteins are there to become protons? Does it require a rocket scientist to say that? It doesn't. They didn't do the calculation. It's very simple. With a simple thing, you can throw that out. It doesn't matter how many people believe in this. It doesn't matter. Simplest thing. There are only 10 protons. There are not. If you want more protons, solubility product of water has to go up. Solubility product has to go up by breaking of HO bond. HO bond is 460 kilojoules per mole. NADH to O2 Erox couple, that is only 220 kilojoules per mole. How can it be 460 kilojoules per mole? It can't. It is energetically unviable. So, it is something that has come out because of some faulty observations. I will come to you. That accuracy and precision.
I'd like to raise a point. There is a phenomenon called proton hopping. proton hopping. Have you heard of that?
It is Grott's mechanism.
Proton hopping.
Yeah, see, the point is this. This is Mitchell's mechanism is to do with the confined species. What you're talking about is catalysis. You're talking about taking proton from one system and throwing it out. Okay. Now, when you're talking about proton,
Electron transport chain, there is certain systems. There is proton hopping for the transfer of proton through the hydrogen bonded
networks, that's called Grott's mechanism. That's what I'm trying to tell you, Dr. Sharif. That is only for within the system. See, there you cannot break and throw it out. Now, I suppose if these were some reductase, some enzyme, okay, they are lax. If they wanted to do a reaction within them which does not require proton consumption for catalysis, there is a product formation that requires intermediate requirement of protons. Acid-base catalysis requires protons that can easily be getting. So now, what you are talking about is, suppose this is the active site. From solvent, it comes it comes to the reaction site. I know Grott's mechanism. That's what you are saying. That doesn't require. See, what you are talking about is in this whole system there is only 10 protons. Now, if you want more protons than 10, there are 10 lakh or 10,000 proteins. You are pumping protons out of the system. You can have this 10 lakh proteins to work with protons, 10 lakh protons within the system. But you are throwing protons out of the system. For that you have to break water bonds. And that requires 460 kilojoules per mole. I don't think you know what that requires. That Grott's mechanism, what you are talking about, it is for catalysis within the system, not to throw protons out of the system. To throw protons out of the system, water will have to break and give more protons. Because this, like Mitchell's concept is, it's a confined system.
In proton hopping, you can transfer the proton from one side of the membrane to the other side.
That requires breaking. No, that they did not make the calculation. How many protons were there? For that only there are 10 protons. They did not make the calculation.
Hypothesis postulated in 1961 by Mitchell.
Yes, and that they did not make a simple calculation. There are less than 10 protons. Now, if it is a confined system, Mitchell's is a confined system. You must understand. The gradient is supposed to work by throwing protons out. Now, the simple thing, you just imagine. What is Mitchell's next thing? Just imagine. At a given point, Mitchell's hypothesis requires lakhs of protons in the system and no protons in the system also. Do you understand that? Protons should be thrown out so that there will be gradient. And do you understand? At that time, you should not have protons and you should have protons also. I will tell you, this is very interesting. You will see. Let me give you some more simple evidence and then we will come to that. Now,
protons are coming from the splitting of water or from the substrate?
No, it doesn't. See, my mechanism doesn't require proton splitting at all. Their mechanism did not have an account for protons. See, if they wanted protons to be pumped out, they should have protons. My argument is, you don't have protons to pump out, first of all. You see, what they showed is, you know why Jagendorf's experiment and Racker's experiment, what they showed is, they took mitochondria, they put acidic pH out, they incubated the pH, the mitochondria at 8 pH, they took a 4 or acidic pH outside, and when the protons moved in, they saw ATP formation. And that is the reason they said this could be one of the strongest evidence. But that is a very different thing, and you will see why it is explained. Because it is an equilibrium based synthesis. I will say. No, I will come to that. I'm coming to that. Now, you see, very simple. Now here, the electron transport chain requires 24 redox active participates in 17 proteins and small molecules. Okay, so and 150 nanometers it has to go, and the kinetics doesn't add up. Okay, the inherently some of the steps that are shown, they cannot be part. Suppose if I am coming from here to there, if the overall reaction is 10 to the power 3 per second. If from here to here it is 10 to the power 2 per second, can this be a part of the overall reaction scheme? It cannot be, because this is inherently lower than that. There are so many things here. You see, when a simple experiment was done in complex 4, there are 2 hemes in complex 4. Okay? Now in that complex 4, when you do anoxic electron transfer between those 2 hemes, the rate was less than 1 per second. You double oxygen, you get 1000 per second. So what does oxygen? Oxygen is the carrier of electrons. That's what. So that's the idea you get. So, and if you see, I don't know how detailed you have gone into it, there's something called as a Q cycle. This is the most finicky reaction that you would have ever seen. It will require a molecule of QH2, Q, so many things to binding, cytochrome C, all that to bind at the same time. The protein should give electron to QH2 and Q at the same time. It doesn't make sense, other than it doesn't make thermodynamic sense, it doesn't make kinetic sense, it doesn't make probability sense. It's only for an explanation part. In real life, reactions are bimolecular, thermolecular, maximum in terms of biological process. Biological processes are shown to have multiple, as biologists we know, the proteins recruit a factor, then another factor, but they are high affinity systems. These are not like that. Okay? So this is the argument debunking ETC. This is the simplest argument against thermodynamics argument. If you simply you take, one molecule of NADH is known to give more than 3 molecules of ATP. So that gives a 48% efficiency. If you take 35 kilojoules mole per term for ATP formation and transmembrane proton coming as 22 kilojoules per mole, then you only have 40% efficiency. So a 40% efficiency.
Pre-ATP is 2.5 exactly.
No, that is precisely. That 2.5 is given by John Walker's DNase paper, and I have argued against that. No, that guy is the guy who is trying to manage things. And if you see plugin, and in my paper there have been at least 20 papers in history. If you see, the title says the yield is more than 3.5. We have repeatedly found. So John Walker is deliberately trying to suppress it. So it's very simple. Evidence is already there. Okay, and then even if you see 40, 45, whatever, the overall glucose to ATP formation, that itself is 39%. What about glycolysis, all those process, transport? There is no window for all that. And then if you see this argument, this will take some time for you to focus. If you are interested, I can talk to you about it. And now past 1 hour, so I want to slowly get to the last part of my talk. So there are absolutely no quantitative justifications for the current hypothesis at all. Will I have a... Yeah. So, let's buy the hypothesis. Let's model the system. Let's put the Nernst equation. Let's give whatever gradient that he wanted. Start with whatever you want. You put. Even then, you won't get the values that is required. See, they have shown experimentally 180 millivolt is required when you see ATP synthesis. Now, to show that 180, you need 90% of the protons pumped from inside out. Now, you imagine now, you have proton pumps. Now they are pumping. How can they pump out like that? Now our heart pumps out. Why? There are fibers in our heart. They pump synchronously, right or not? So all of them can pump out together. These proton pumps, they are all located here and there. How can they pump synchronously? So if one pumps now, another pumps later, the proton has gone and come back. So where does the gradient build? It is just a mirage that was sold. This quantitative analysis modeling also doesn't support ATP synthesis. Now you see, structurally, this puppy here, this unit is supposed to hold on to the membrane and rotate at the same time. Okay, can you imagine that forming on a natural way? Of course, there have been single molecule experiments which have shown. But in a circus ring, you can take a lion, make it sit like that and then do a lot of things. But that doesn't mean that it will have any physiological relevance. I have argued against this in so many ways. See, very simple. You can take a fruit, okay, put it into a mixie. You will get juice and pulp at the other end. You take juice and fruit, try to invert it. Would you get a fruit? No. That is what the current hypothesis wants. It's completely against thermodynamics. The ATPase enzyme is ATPase. It doesn't have ATP synthesis. But in a closed system, when the ATPase is present, then you observe ATP synthesis. That is what is seen. Now that can be inferred in very, very different ways. Okay? That is something that we have to see. Please see this. It's a very interesting picture. Have you heard of Esther? See this. You see this? Water falls from the second floor and you try to see the progression of the flow of water. It seems to be going downhill all the time. And then it comes and falls again, driving the wheel. Is this possible? Is this possible? It's not. But that's what Mitchell's idea is. Protons are pumped inside to out, like that. When there are no protons, and then thermodynamically it doesn't make sense, modeling doesn't make sense, it doesn't make sense at all. So, it's like a Mobius strip that was put in place. You know what a Mobius strip is? So, not just me. There have been so many people who have, if you see, this man, Slater, E.C. Slater, was the editor of Biochemica Biophysica Acta, the leading journal in bioenergetics area. He opposed Peter Mitchell's ideas for a lifetime. And he had called to scientists to come up with some alternate explanation. I want you, my dear students, if you, you're not my students, sorry, please see this name, Lim. He's 94, 95 years old. But I feel so sorry for him. In 1960s, he put forward a very fundamental hypothesis called association induction hypothesis. You won't know about it. He has been sidelined, but he was lucky because, Raymond Damadian, the guy who, MRI, the big MRI instrument, those people who own that instrument, that's Patron took this guy, gave him a lab, and he has got a financial way to sustain. So that way he's lucky. So, but this guy has some very radically different ideas about cell, how cell works, how cell homeostasis is. So, please, if you are interested in real ideas, read him. Okay? So, these are the other people who have doubted. Therefore, I have conclusively debunked this hypothesis. And in fact, I have projected so many experiments also which you can do, which can show that my hypothesis is right and their hypothesis is wrong. I cannot say that my hypothesis is right because any time a new data piece comes around that challenges my hypothesis, it stands to be falsified. But definitely you can prove that this is wrong. So, now my murburn concept was established for peroxidases and P450s. Now I am coming to the third and final part of my talk, debunked, after the other concept is debunked. Here, now once again we are coming to the initial part, what I said now. You see, if you put an equation of a circle, say for the circumference of an equation of a circle, what is that? 2 pi, right? Very good. Now, if you get data that fall in this, all the data that fall in this, it's a beautiful circle, fantastic. But some data, if they fall like that, you can call this an artifact. Okay? And then you can choose to define the overall system with this. Now the accuracy and all that are creeping in now again. Now I can choose to disregard and still see circles. You see here? I can still see circles if I want to. If I want to, I can still see circles. Here is a Michaelis-Menten curve. I have taken a Michaelis-Menten curve. I have taken 10 points. Simulation. Absolute R squared 1. I have used this equation to get this Michaelis-Menten curve. And then that same hyperbola I have put in a parabola. You see the fit. R squared is 0.994. Okay. Now you see totally disconnected theory. You put it on one phase exponential equation, you get R squared is equal to 0.99. Put it in two phase exponential, you get it R squared is equal to 1. So seeing is not necessarily believing. You know, a dog licks water, right? Laps up water. You know how it does it? You will think it does it like that, right? Like we lick things. No. What it does is, it uses the bottom side to scoop the water into the lower part of the jaw. Do you understand? We thought that, it is the sun that goes around Earth. So many experiments, so many observations. Accuracy, it is always on the scientist. So, it doesn't matter how many people say certain things. If you start questioning, if you start getting even one data point, you are going to be the experimenter. When I was working with Lowell, I was just doing one experiment. Chloroperoxidase, azide. I was adding azide. I added millimolar azide. Stop. Micromolar azide. Boom! The reaction went up. I was thinking, something wrong? Something wrong? I explored that one single observation. If you try to fit that observation into this, saying that it's an artifact, you will get it if you want to see it. You understand? But I pursued that one single observation. And that one single observation, I have come to this point where I can explain xenobiotic metabolism, the chloroperoxidase, explains the recycling of plant lignocellulosic materials. They are supposed to go through the chloroperoxidase mechanism. The metabolism of drugs. Now, currently, I am 3/4 into debunking the mechanism of photosynthesis now. Okay? My mechanism also explains photosynthesis. You will see at the end of it, what are the... Now you see, what are the similarities between the liver system and mitochondrial system? Overall reaction, same. Electron donor, NADPH, NADH. Acceptor is supposed to be molecular oxygen. Flavoproteins, hemoproteins, low flavoproteins, high hemoprotein. Same. Cytochrome B5, cytochrome C. Reaction center stage, phospholipid interface vesicles. Same. Final product, similar. Hydroxylated, ADP is phosphorylated. Multi-enzymatic, ordered sequential scheme, multi-substrate, multi-product. Or same kind of reaction setup in both. In terms of phenomenology and mechanism, electron transfers, partitioning, protons, thermodynamic drive, substrate binding, water formation, Ross involvement, inhibitors, cyanide, uncouplers, stoichiometry, all these explanations were very similar in the xenobiotic metabolism. I have conclusively changed that. So what I am trying to show here is, how come murburn concept can be relevant for this system? So, similarities between established system and this system. Dr. Sharif, I am coming to your question. What is your evidence? What are your arguments? So, similarities. First, establishing some kind of a, case for that. Then, non-specificity. Redox systems. You see, ETC you study by giving so many diverse kinds of electron donors and acceptors. So, there is no real specificity. Anaerobic system, you give oxygen. They die. Why? Because oxygen shunts everything. If it is through a very specific kind of electron donor exception, then you would not, it is not supposed to be like that. So there is no specificity. So electron transfers are dependent on more fundamental aspects and not affinities per se. My hypothesis is very simple. Superoxide is formed and peroxide is formed by the complex 1 and complex 2. They are stabilized and further channelized by complex 4 and complex 3 and things like that. And there is a zone here. In this zone, there is a region where diffusible radicals are present. And this area, there is high levels of millimolar levels of phosphate.
Diffusion through the membrane?
No, not diffusion through the membrane. Here are the, now you see, this is the cross section of the membrane. This is the complexes that are protruding out of the membrane. Now, here, the electrons are supposed to go through the wires along this, right? Complex 1. There are redox centers here. Now, what I am saying is here superoxide and all that is produced here. Okay. Now, here phosphate is present. Now, the superoxide, these radicals here, charge the phosphate. And phosphate and ADP, then very simple reaction.
Diffusion from where to where?
No, there is no diffusion. There is diffusion. Here you see, here.
And diffusion is independent of any energy. It doesn't require any energy. It needs it moves from a higher concentration range, concentration region to a lower concentration.
Yeah, here there is present a lot of radicals here, and they just attack phosphate and charge and ADP synthesized. It's a very simple reaction. Now here radicals are produced here in this area. In my hypothesis, cytochrome C is present as a 1 electron scavenger in this intermembrane space. This is a simple reaction dome. This is the cover of the reactor. This is like a nuclear reactor. The analogy in my reaction system is like that of a nuclear reactor. Okay? It is no pump, nothing like that. Here it's a very simple chemical reaction. Here these complex 1 and complex 2 flavoenzymes, they produce reactive oxygen species. Those reactive oxygen species react with phosphate, charge the phosphate, and ADP reacts with phosphate. Now this is a very simple reaction model, and this explains why enzyme based intermediates were not discovered, because this is not an enzyme based reaction. That's why. And you see, I have already proven it in 2 systems.
What is the function of these complexes?
That's what I said. The complexes are there to
They are
there to generate reactive oxygen species and stabilize.
enzymatic reaction.
Pardon?
That is an enzymatic reaction. Isn't it?
Yes. They are there to present. They are currently diffusing reactive oxygen species are seen as side reactants. They are seen as unwanted stuff, right? They are seen as challenge to life. What I am saying is a paradigm shift in biochemistry. What I am saying is, it depends upon where they are present. It is like knife. You cannot say knife is bad. If you use knife carelessly, knife is bad. This I have shown in 2 systems that when diffusible reactive oxygen species are present.
The concept is the superoxide anion is produced because of the leakage of electron from the electron transport chain. That is the concept.
Correct. I am saying it is not a leakage.
That concept is holding good for your hypothesis.
I am not saying it is the leakage. I am saying it is the primary function. It is not leakage. That is why it is there. See, you see, the simplest evidence. You ask me for evidence. This is the simplest evidence. When ATP is synthesized, 180 millivolt is the experimental point. When reactive oxygen species are.
But this superoxide anion is highly toxic to the membrane, injurious to the biomembrane. Then so many molecules are supposed they are formed. What happens to the biomembrane?
Lovely. This is what. Okay. Very simple analogy I give you. Take a cloth. Take a cloth. Dip it in oil. Burn the oil. Burn the cloth. What will burn first? Cloth is untouched. The oil burns. Cloth, nothing happens to the cloth. That is the mechanism. That is why they call it murburn constant. Now, it's a very powerful analogy. You should understand. That's why we need unsaturated fatty acids. That's why cardiolipin is found in mitochondria. Cardiolipin has high amount of negative charges. It pushes the superoxide into this Murzone. My name is Murali, so I deliberately kept it as Mur. Yeah. So I'm letting you know I have. Finders keepers. What the heck? So I call it Murzone. I call these enzymes Murzymes, and so better deal with it. I die, but this will remain. I'm very confident. So yeah. So you see the structures. You see. If my hypothesis is correct, you see, what is the probability that a car evolves or a dam evolves on its own? Something has to be counted. But then my model is like a nuclear reactor model, stochastic control. In history of the planet Earth, there is a naturally formed nuclear reactor. In Gabon, in Africa, approximately 2.5 billion years ago, it worked for hundreds of thousands of years. So, a natural nuclear reactor can spontaneously form. My mechanism is very spontaneous.
May I ask you
Yeah, please.
Suppose you isolate the mitochondria and suspend in a suitable medium, add a lot of superoxide anion, could you get ATP synthesized?
That is one of the experiments that I have projected. Very good question. For confirming. In fact, it's already been confirmed by an indirect way. I'll come to that. Now, your question is very good. Let me tell you. Now, in P450 system, very, very good question. Now, in cytochrome P450 system, what they did is, when they took, in the reaction system, they would put catalase and superoxide dismutase. And the reaction would go on perfectly fine. Now, they said, if superoxide is involved, how could this reaction go? Now, I showed them very simple. My work is all published, you can see. You take vitamin C, it doesn't inhibit the P450 reaction. Okay? You put a fatty acid chain to vitamin C, bam! Zero activity. You take vitamin E, which has got a fatty acid chain, it's not a fatty isoprenoid chain, very correct. Fatty chain, hydrophobic chain. It inhibits. Trolox, which is a substituent which has the hydrophobic part taken off, it doesn't inhibit. Now, superoxide dismutase is a polar aqueous soluble enzyme. See, the point is how quickly it is produced, how quickly it is utilized. If there is this reactive oxygen species, if they don't find others, they react among themselves. That's the beauty of the system. I have shown this in chloroperoxidase reaction. These two systems, I have shown all these doubts that you are having. I have clarified all that. Very, very good doubts. Very, very good doubts. And these are only doubts with respect to aesthetics part. Mine is with respect to the kinetics, mechanics, and thermodynamics. See, a reaction, the current mechanism doesn't explain kinetics, mechanics, thermodynamics, energetics. As people of science, we cannot have irrational ideas. Mine is a totally rational idea, but it has got a history of aesthetics to cover. But if you see this is very good strong evidence. When reactive oxygen species are present, only when they are formed, ATP is formed. Direct correlation. Without ROS, there is no ATP. And then there are so many other reaction systems where the reaction that you are talking about, the Racker example, for example, Racker showed with rhodopsin, they just took rhodopsin and they took ATPase and they used light and they saw.
It is rhodopsin which has got this proton pumping.
Yeah, proton pumping, that is what they call. Yeah. So now what I am saying is it's not proton pumping per se. It is rhodopsin produces reactive oxygen species. Rhodopsin produces reactive oxygen species. It has been shown. And the system in which that experiment was done, it had flavoenzymes, produces plenty of reactive oxygen species. And in those systems, they saw that several amino acid residues with serine, they also got phosphorylated. you know the structure of serine, students? Serine has a hydroxyl group. Hydroxyl group can get phosphorylated NACA. ADP has hydroxyl group that also gets phosphorylated. The reaction chemistry is the same. So many things. Now you see the distribution density. It is a randomly distributed one. Complex 1, complex 3, complex 4, complex 5. See the relative distribution. It is beautifully agreeing with my projected hypothesis.
What is the concentration of this superoxide anion in the mitochondria?
Well, Dr. Sharif, we have no.
You calculated the
No, this
concentration required. But what about this superoxide anion concentration?
This is a very relevant question. The problem with this aspect is, in situ, at the site of origin, how much is produced, how much is present, you don't know. We don't know. We cannot calculate. Now, I'll tell you the particular reason. I'll tell you one second. Let me tell you. When I put HRP, which is an enzyme transmembrane segment, it has a transmembrane segment. It completely inhibits the CYP activity. Okay. So, the fact that superoxide is involved is conclusively proven. It is not membrane protein electron transfer. Because I have shown the two proteins separated by a dialysis membrane and still electron transfer occurs. So it is only through a diffusible species.
That is because of the difference in the oxidation reduction potential.
No. See, currently Rudolf Marcus theory says electron transfer among proteins, within proteins, is through electron tunneling. And that can be through interprotein or intraprotein. Okay? One protein comes, contacts, in contact, and that is the reason, affinity. Because if you had chaotic reactions, electrons being transferred just like that, then you would have the same problem.
And the electron binding to the protein may be changing the conformation of the protein, and maybe giving, because of the change of the conformation.
This argument cannot explain how I can get electron transferred by protein separated with dialysis membrane. Two, with respect to kinetics, it is completely impossible. If you do the simple calculation with respect to mobilities of the proteins on lipid membranes, it cannot add up. It comes 3 to 5 orders lower than the observed reaction rates. It cannot.
Dialysis membrane use?
These are ordinary dialysis membrane cellulose. Cellophane. That is published in RC Advances. So, now the concentration of superoxide, it is not possible to determine at this juncture. The point is, I have shown, what I did is, I took potassium superoxide, I stabilized potassium superoxide in crown and dimethyl ether, and then I gave it to CYP without. Can you please listen to this this argument? I gave superoxide with stabilized in crown and dimethyl ether. Crown ether. It can have finite lifetime in water. So when superoxide is given like that, I get exactly CYP reactions. I don't need the reductase. So there is no electron pumping like that. It's just superoxide that is active. I have shown experimentally, very clear mechanistic conclusive, I have got theoretical mechanistic conclusive proof.
How do you publish the superoxide anion is responsible?
Yes. Not in the mitochondrial system, but in the P450 system. It is the exactly simple same system, but in a different model. So now, okay, now my time is almost 1.5 hours. I asked for this only. So these are the different equations and all that. So all this I said. So basically, my hypothesis agrees with the Occam's razor, one of the most fundamental aspects, Charles Darwin's evolution theory. In fact, it explains why cells die, how cells die, in fact, in terms of energetics. Why do you observe apoptosis with leaching of cytochrome C? Because the Ross machinery is out open and the whole system is
gone, so it has to better commit suicide, or else the whole animal or organism will go. So many aspects it explains. Distribution of proteins, bimembrane, heat generation. How does brown adipose tissue generate heat? Because they have positively charged amino acids in their loops, so they can dismutate superoxide like that. So toxicity of cyanide, so many redox potentials, all that. So yeah, so basically whole textbooks, everything has to change. Why do we need oxygen? We need oxygen to form radicals at the phospholipid interface. Oxygen is like the Batman character, or Dr. Jekyll, Mr. Hyde, if you have heard of it. This two-face, it is subject to a lot of chaos, but chaos and randomness is what life has evolved from. And it is wonderfully, you can demonstrate with these systems. I have shown that non-active site reactions can give selective reactions, can give specific reactions. And that's what murburn concept is. So it's a paradigm shift in biochemistry, and you see, if you know Thomas Kuhn's or Michael Polanyi, whoever you follow, the way in which science changes. A new idea takes its own time, it goes through a cycle. But as far as accuracy, scope, simplicity, utility, all that is concerned, my hypothesis has got a lot of scope to explain a lot of things. Only thing is consistency aspect, because what one system, if you do an experiment and if you do an experiment, a slight change in the variables can give difference in values. And that is the inherent nature of the radical. So that is consistency. It can cause a problem with consistency, but the theory itself advocates consistency. So now it is poised to explain photosynthesis, overall bioenergetics, a lot of aspects of cellular homeostasis. I plan to use Link's hypothesis, merge mine with the current understanding to get a major idea, and then this sensing stimuli, how do so many mechanical stimuli get transferred into biochemical stimuli? We are biochemical systems. We have only one force that we can use, that is electromagnetic force. So we need chemical stimulus. Suppose somebody touches us, we hear something. How do these signals get transferred? I say that it is all through this molecule, unbound ion, and radical interaction, because only radicals can give quick reactions, and our systems have evolved to use these mechanisms, and we currently see it as only bad. No, they are not just bad. Don't see it as black and bad. It is a paradigm shift. You need to see it as bad in certain circumstances, not bad every day. That's the message of this talk. Other than that, this is the very simple cartoon that I always put after my talk. Please take a minute to go through it, particularly the students. It shows you exploration is a very important part of science. Sadly, modern day science only uses hypotheses. You start off with hypotheses. You try to fit your data into hypotheses, and that's sometimes not necessarily a good thing. And sometimes it's important to go on exploring and questioning and going beyond available paradigms. Thank you so much for your patient listening. Thank you so much for your attendance. And I do not apologize for my passionate nature. That's who I am. But this kind of irritates people whenever I give a talk, because most of the senior people, what, we are some fools or what? You are only the right person. Yes. In this particular thing, you prove me wrong. I am challenging you openly. I have got very simple arguments. Very, very simple arguments. There are no protons. It is energetically improbable. Oxygen cannot stay wedded to complex 4. The kinetics don't add up. The energetics don't add up. Thermodynamics don't speak sense. Nothing speaks. Biology doesn't speak. Evolution doesn't speak. It's all for me. Only thing that is not for my case is me saying it's me, and I have no other people who are going to support me as of now. So, if you are interested, if you have got the resources, and even if you are doing disconnected agenda, please, I beg you, students at least, have the inclination to question. Questioning is very important. And that's why a simple guy like me can stand. And today I have challenged so many paradigms, and nobody has an answer. They don't accept it, but then they'll come back to accept it. They have no other way. I've got all the arguments. It's just that if you see the first slide, I was talking about this before the talk commenced. A new scientific truth does not triumph by convincing its opponents and making them see the light, but rather because its opponents eventually die and a new generation familiarizes with it. This is said by who? Max Planck. You know what Max Planck did? Quantized. Everything was seen in continuum. He brought quantization. One of the most radical ideas. Okay? Now what did he do? When Richard Feynman, he put forward the quantum electrodynamics and quantum mechanics and all that, he opposed it to Neil and Clark. He said, this is all wrong. Einstein said wrong. But then that was right. That's how it is. So, people can't say why I am wrong. I can say 100 reasons why they are wrong. So that's why that image that I showed, the circle, the hyperbola, all that, the accuracy, that argument, all that, it's all depending upon you. If you can, today, if you have a lot of followers, you can make anybody believe anything. So I'm not going into that. You had asked me to.
On a lighter note,
Yeah, please.
Calvin's Hobbes is an imaginary Bengal tiger.
Yeah, I know. I know. It happens to be my most favorite comics. I have all the collections. But I still, you see, comics, it's a wonderful world. You learn so many things from comics. I grew up with comics. Even now, I love comics. I love animated movies. It's a wonderful world. It's a wonderful philosophy that Calvin adopts. Please read those comics. Wonderful comics. You have read them? Lovely. Royal Bengal Tiger. Yes, it's a stuffed Royal Bengal Tiger. Yeah.